Welcome to Azad Bioacademy. Today, I will talk about action potential. As you know, action potential is a very vast thing. So, before going to detail, I will just give you some basic of action potential, membrane potential, and some ion channels, receptors, which are basically connected to the action potential. Okay, so firstly, different types of ions, ion channels, receptors, and then action potential conduction, and finally refractory period. So the video will be somewhat longer. Okay, so stay with me. So come to the first part of this action potential, that is different types of ion channels which are basically present. As our topic is action potential, so we have to consider the action potential are basically responsible for some skeletal muscles, cardiac muscles, smooth muscles, and we can say the nerve cells. Okay, and membrane potential is applicable for each and every cell. So as our topic is action potential, so we'll consider as a nerve cell for the details discussion. Okay, so if I just consider nerve cell uh, on which I will discuss the action potential, so how many numbers of ion channels are there and what are their roles. Okay, so firstly, if you think this is the membrane of nerve cells, so how many ion channels are there? So firstly, uh, if I just discuss one after another stepwise. So firstly, I will talk about the channel, that is leak channel. Okay, leak channel. You know, nerve cell has different types of leak channels: sodium channel, potassium channel, and chloride channel. Some leak channel. What does it mean, leak channel? Leak channel means is the protein channel which is always open. Okay, but in nerve cell, you know, the leak channels is basically considered as a potassium channel. Okay, potassium channel. Why it is considered as a potassium channel? Because leak channel, potassium exit through the leak channel is frequently happens in the nerve cell. Because you know the molecular size of sodium is somewhat larger than potassium, so frequently sodium cannot enter or exit through the leak sodium channel as chloride channel. So leak channel, through the leak channel, basically potassium exit. Okay, so one of the major ion channel on the surface of the membrane of the nerve cell, that is potassium channel. Okay, that can be considered as a leak channel. Second one is sodium potassium ATPase. ATPase. This is also called sodium potassium ATPase or sodium potassium pump. You can say this is the exchanger channel. Okay, and this channel is basically activated by the energy, that is ATPase. Okay, so that will use the ATP for the energy production, and that will contribute for the conduction of sodium and potassium through the channel. That means through this channel, 3 sodium will exit and 2 potassium will enter. Okay, so if I just think this is the number 1, that is leak channel, and this channel is, you can say this is just as I consider the ATPase. So this is the channel through which 3 sodium will exit and 2 potassium will enter. Okay, potassium will enter, and basically here the leak channel, so potassium will exit, which is leak channel, that is always open channel. Okay, so you can just consider sodium potassium ATPase channel as like a sodium potassium pump. Okay, and third one is you can consider as a mechanical sodium channel. Okay, mechanical sodium channel. What does it mean, mechanical sodium channel? This is very sensitive. A mechanical force, that is, you can say touch, you can say pressure, and that is very sensitive. That sodium channel is present on the surface of the membrane of the nerve cell, just like that. In general, this is not open as like the leak channel, but it needs some mechanical forces like touch, pressure, and then it will open, and through this sodium will enter. But remember, this mechanical sodium channel, you can say touch sensitive sodium channel, is not like the voltage gated ion channel. Okay, remember it. So you can say the mechanical sodium channel, or you can say this is another one is touch sensitive sodium channel. Okay, sodium channel. Okay, fourth one is you can say voltage gated ion channel. That is voltage gated ion channel. Ion channels. So voltage gated ion channels means the channels will open or close by the influence of voltage difference, that is the potential changes. Okay, so that is the voltage gated ion channels. You can say voltage gated ion channels are sodium channel, potassium channel, chloride channel, like that. Okay, here in nerve cell, voltage gated sodium channel and potassium channel are very much prominent than the chloride channel. And the fifth one is ligand gated ion channel. Ligand gated ion channel. That means ligands, endogenous or exogenous ligands, are needed to open the channel or close the channel. That means ligand is a must to control the ion channel. That can be ligand gated ion channel, that can be ligand gated sodium channel, ligand gated chloride channel, ligand gated potassium channel, like that. Okay, so basically here ligand gated sodium, ligand gated calcium, or ligand gated chloride channel are there. Okay, so these types of ion channels are basically present in the nerve cell. Okay, so the leak channel, what would be the role of leak channel, what would be the role of sodium potassium pump, mechanical sodium channel, voltage gated channel, and ligand gated ion channels. Just I will just discuss the roles of these channels regarding the action potential. Okay, so before going to the action potential, we should think about their roles, the basic roles. There is leak channel, basically exit through this channel, basically exit the potassium, and that will maintain the graded potential as well as basically graded potential. So I will discuss later on. Sodium potassium pump, that will basically maintain the resting potential. Mechanical sodium channel, that is very much sensitive to the touch or pressure, but it will not as like as a voltage gated sodium channel, that will work below the threshold level, that is in sub threshold level or degraded potential. And voltage gated ion channel is very much important because that channel is basically contribute for the action potential. Okay, and the ligand gated ion channels, you know, the ligands are basically neurotransmitters, hormones, and other biochemicals which basically work on the specific receptor, and that receptor will control the ion channels. Okay, so this is the basic of ion channels which are basically responsible for the action potential. Okay, so now we will go for details. Action potential. Okay. So now come to the main topic is action potential. So actually, before going to the action potential, what is the potential means? What is the difference? Different membrane potential. What is it? Actually, the potential difference inside and outside of the membrane of the each and every cell is very much important. If you think that a signal transduction pathway is very much important, the membrane potential. That means each and every cell, the membrane potential is present. That is always it is in polarized state. That means the ion exchange or the amount of ion inside and outside is always not in the equivalent state. Always in you can say the difference inside and outside. Okay, so you can say that is membrane potential. That means outside and inside has the potential difference. That means in polarized state. Okay, so when it will be considered as action potential. So always membrane potential is always present and present each and every cell, which are their normal function for the signal transduction and other some releasing factor of some internal content. Okay, so come to the point now. So what would be the action potential? Action potential you can consider as like when the potential difference or potential will just travel. That means when it will be in action, that is action potential. The potential energy is very much important to make some changes within the muscle, within the cell. That is the action potential. That means it will travel and it will just make a complete event. That is the action potential. And membrane potential is just ion exchange. That's like electricity. That means electricity is the flow of electron or the flow ion exchange through the membrane. That is the electricity. So action potential, when the electrical event will just travel. Okay, so that is the action potential, and it's very much important for particular cell and muscle. If you think about the nerve cell, the action potential is very much important, because without action potential, neurotransmitter will never release. Okay, suppose if you think about your cardiac muscle, without action potential, your heart muscle will never contract or never relax. If you think about your skeletal muscle, that will never contract or relax without the help of action potential. And as like a smooth muscle, your blood vessels like that, of course action potential is very much important. And some glands also use action potential for their contribution, as like you can say the beta cell for the release of insulin. Here some action role of action potential. Okay, so come to the point, action potential. So action potential, when I will just discuss action potential, we have to discuss a complete event. So I'm just drawing firstly the basic thing. One is your firstly the resting membrane potential, and then it is the threshold potential, and you can say this is the peak level. And so if I just mention here minus 70 millivolt, this is the resting potential, okay, and minus 55 millivolt, you can say this is the threshold level, threshold potential, and here it is the peak point, peak level, that can be plus 30, somewhere 35 millivolts. and in some particular cases that can be maximized in plus 55. This is highly stimulus way. Okay, so this is exceptional. So here that is the peak level. Okay, sorry, peak level. So resting potential and the peak potential. So below the resting potential, another potential is present that can be considered as a potassium equivalent potential. Equivalent potential, and you can mention it, this potential below the resting potential is hyperpolarized state. So I will talk about now one after another. So firstly, without any stimulation, without any touch, or without any activation, the membrane of the nerve cell, the potential difference is minus 70 millivolt greater inside the cell than outside. Okay, suppose if you think this is the nerve cell, and if you just incorporate a microelectrode inside the nerve cell, so you will get in galvanometer minus 70 millivolts inside the cell. That is minus 70 millivolt greater potential or excess potential inside the nerve cell than the outside. Okay, so in resting membrane potential is minus 70 millivolt. In that case, how it will maintain, I will talk about later on. So firstly, this one is minus 70 millivolts resting potential, and this one is your threshold potential, and then it will go up to the peak level, then again it will just come down, and again it will reach to the resting potential, and then it can reach below the resting potential, and this way. Okay, so you can just give some name of the event. Okay, this is the resting membrane potential, how it will be maintained, the resting membrane potential, and before reaching to the threshold potential, the potential difference is there, and what would be the terminology for that potential changes, and what would be the terminology for that region and that region. Okay, so now I will talk about that. So firstly, so without any stimulation, suppose your nerve is in rest, so no activation. So when you will activate, then what will happen? The activation or stimulation must be optimum, because nerve cell follow all or none law in regarding action potential. That means stimulation must be optimum, and that will create action potential that will reach the threshold level, otherwise it will not work at all. Okay, so before threshold level, by some your leak channel or by some your mechanical sodium channel, here some potential changes may happen. And you know, already I said the membrane is always in polarized state. That means some extent ion is already just already enter or exit. Okay, so resting membrane potential is minus 70, and here when nerve is just slightly touched or pressurized, and it will just can increase the potential towards the positive. Okay, minus 72, like that, like that. It will just if you think, you will touch the membrane of the nerve cell, and then mechanical sodium channel will open and some sodium will just enter. Then what will happen? Now, when sodium will enter, the potential change will occur towards the positive current, okay, but it will not reach to the threshold. So actually this current or this potential changes is meaningless. So what is that? These are the graded potential, okay. You can say these are graded potential, or you can say these are this is basically sub-threshold potential. Sub-threshold potential, because the stimulus is somewhat less extent to reach the threshold. That means some touch or some pressure or some mechanical just mechanical stimulation that will open the touch sensitive sodium channel, and some sodium will enter, and then it will change some potential, and that will never reach the threshold, and that is the sub threshold. And that will, when the potential change will occur towards the positivity below the threshold, and that will happen by the entry of sodium, and again it will minimize to the resting potential, like that, okay. So this is basically happened by the leak potassium channel. Already I said potassium channel is always open, leak potassium channel. So that through this potassium, so some potassium will exit. So sodium will enter by the mechanical sodium channel, and potassium will exit through the leak channel, and it will balance. That means some extent it will increase and then decrease, then increase, then decrease, but that will vanish, okay. So this is the graded potential or sub-threshold potential, okay. Then what will happen? If the stimulus is optimum level, if the stimulus is sufficient to create action potential, that means that will reach the threshold potential, then what will happen? Okay, so without reaching the threshold potential, action potential never start. Okay, so here when you will just activate or stimulate a nerve. Suppose this is the nerve cell. When you will just stimulate live through the fire, then what will happen? The potential change will quickly reach to the threshold potential, and when threshold potential will reach, then the voltage gated channels will open. The voltage gated channel already I discussed, that is voltage gated sodium channel and potassium channel is very much important uh, to initiate the action potential and to maintain the action potential, okay. So from here the voltage gated sodium channel will open through this, okay. So actually I'm just discussing different parameters, but in resting state, what is the membrane condition of the potential or ions inside and outside? That is already I missed out. So just I'm just drawing here. In the resting membrane, resting potential, that would be the right place to just draw a resting membrane intensity of the action potential, sorry, intensity of the potential difference. This is the membrane. In normal, that is in resting state, outside of the nerve cell, sodium is very rich. Sodium, okay. That means outside of the nerve cell, sodium are very rich outside of the cell. That is potassium. You can say that is potassium, and some chlorines are there. Chloride ions are there. Okay, so sodium is very rich. Okay, and inside the cell, potassium is very rich. Potassium is very rich. Okay, and some sodium there. Okay, so you can say in normal resting state, outside of the membrane, sodium is very rich than the inside, and inside the cell, potassium is very rich. Okay, so the overall intensity of the potential is the membrane. Outside of the membrane, the current intensity is positive, and inside is negative. Why it is like that? If you think sodium is also positive charge and potassium is also positive charge, so why the sign of the electricity outside of the cell is positive, or intensity of the potential inside the negative? Why? Because the number of sodium is higher or very larger than inside the potassium. That means number of potassium is very less as compared to the number of sodium outside of the cell. Okay, so positivity is greater than inside. So this is positive. Another thing is that within the nerve cell, some negative factors, negative charge are there. That means if you consider as a protein is negatively charged, so that will maintain the negative intensity of the potential inside the cell. Okay, so here I just want to say in resting state, outside of the cell, sodium is very rich. Number of sodium is very higher than inside, and the intensity of the electricity outside of the cell is positive and inside the cell is negative. But the sign of positive and negative does not mean positive current or negative current. Electricity has no positive or negative. Actually, it means the intensity of the potential changes. Okay, so now come to the point. So here, when the threshold stimulus is very strong and it will reach the threshold level, then what will happen? The voltage gated like that. You can say there is a voltage signal like that. Voltage gated sodium channel will open. So what will happen? Voltage gated sodium channel will open. So voltage sodium channel has some gate, the activation gate, inactivation gate, but I will not go to the details about the voltage gated sodium channel, but I will just focus on the role of the sodium channel, voltage gated sodium channel in action potential. So what will happen when the threshold level will reach by the stimulation, then voltage gated sodium channel will open, and what will happen as sodium is more in outside of the cell, then that will enter through the sodium channel, voltage gated sodium channel. That means sodium will enter. Sodium will enter. Sodium. Okay, and the potential changes will just happen toward the positivity. That means that current will be called as depolarization. Depolarization. Okay, that means depolarization potential or depolarization current will produce by the entry of sodium, because at the threshold level, voltage gated sodium channel will open and rush of sodium will occur inside the cell from the outside, because through the voltage gated sodium channel, sodium will enter through this channel and it will cause the potential changes toward the positive way, and that will make depolarization. That means depolarization will happen through the entry of sodium through the voltage gated sodium channel. That's it. Okay. And what will happen? It will just reach to the peak level. That means maximum positive potential. That is the peak point. Okay, and that can be 30, somewhere 35 it is mentioned, and it will be maximum, that is plus 55 millivolt. That can be considered as a sodium equivalent potential. Okay, so after reaching to the peak level, then what will happen? Instantly the sodium channel will close. Okay, sodium channel will close, and then what will happen? The voltage gated potassium channel will open. Voltage gated potassium channel, that is voltage gated sodium channel, that is voltage gated potassium channel. So here voltage gated sodium channel will close, and then voltage gated potassium channel will open. Okay, and as you know, inside the cell, nerve cell, potassium is very number of potassium is very much than the outside. So what will happen? Through this voltage gated potassium, potassium will exit, potassium will efflux. Then what will happen? The intensity of the action potential energy will just decrease, that is, it will just go towards the resting. That means your nerve cell will just try to become, the potential changes try to become a resting level. That means it will just again go to the rest. Okay, so the resting the potential changes will just go toward the resting membrane potential, and that electrical just potential changes is called repolarization. That is called repolarization. Repolarization. So again, simply, how do you say? What does it mean, repolarization? Repolarization means through this voltage gated potassium channel, potassium will exit. And what will happen? When potassium will exit, the potential changes will go towards the resting level, and that is called repolarization. That means repolarization will happen by the exit of potassium through the potassium voltage gated potassium channel. Okay, and it will just reach again to the resting level. So from here to here, this is the repolarization, and from threshold potential to the peak, this is the depolarization, and this is the depolarization, and this is another potential, this is the repolarization. So depolarization will start from here and repolarization will start from here, and it will end in the resting potential. Okay, so this is the depolarization, this is the depolarization, and this is repolarization, and actually this is called action potential. Okay, this is called action potential. Okay, because in that way the potential changes, the potential energy will travel. Action potential. Okay, so this is basically, truly, this is called action potential. So what does it mean, action potential? Action potential means depolarization and repolarization. But that after reaching to the resting membrane potential, that can go backward, or that can just change the way where it will start the potential. So here some some changes will happen below the resting membrane potential, and that is very crucial. That is called hyperpolarization, and this is very much important because it will prevent the backward potential, and it will also maintain the accuracy of the action potential towards the next one. Okay, so this is the hyperpolarization. Hyperpolarization means when your electrical potential change will happen below the resting membrane potential, this below. Okay, so you can say this. Okay, that means from resting membrane potential to potassium equivalent potential. This is the hyperpolarization. So what does it mean, hyperpolarization, and how it will happen? Hyperpolarization will happen through the leak potassium channel, as I discussed earlier. That is leak potassium channel. That means through the leak potassium channel, some potassium will exit, and then it will just go below the resting membrane potential, because at the when it will just reach to the resting membrane potential, the voltage gated potassium channel will close, and you know the leaky potassium channel is always open. So through this channel some potassium will continuously exit, and that will cause hyperpolarization, and it's very important, hyperpolarization. Okay, so this is the hyperpolarization, and that hyperpolarization can happen through another channel, that is ligand gated chloride channel, that will I will discuss when the conduction and the application of the action potential over there. But here basically when action potential repeatedly happen, so uh, in very few milliseconds hyperpolarization can happen as the refractory period. I will just talk about just after this action potential. So here is the uh, hyperpolarization. So that this hyperpolarization will just go just below the resting membrane potential. That is hyperpolarization. So if you think already this is hyperpolarization, and that potential energy is minus 90 millivolt. That is minus 90 millivolt. Okay. Okay, then what will happen? This minus 90 millivolt here already reach the, you can also consider as a potassium equivalent potential. Then what will happen? From this point, the potential change will again reach to the resting. That will again it will go to the resting membrane potential for further stimulation. Okay, so who will maintain this part? That will be maintained by the sodium potassium ATPase pump. Okay, sodium potassium ATPase. Okay, sodium potassium ATPase. This is the touch sensitive sodium channel. This is sodium potassium ATPase pump, and this is voltage gated sodium, this is voltage gated potassium, and the voltage gated potassium, and this is leaky potassium channel. Okay, So already I said here what will happen. The sodium potassium pump that will maintain the resting membrane potential. It will maintain the resting membrane potential, and through this hyperpolarization it will maintain. How? Because through this sodium potassium ATPase pump, 3 sodium will exit and 2 potassium will enter, and that will maintain the resting membrane potential. Okay, so hyperpolarization is a part of action potential or not? So you can consider as a third phase of action potential, hyperpolarization. You can consider hyperpolarization is a part of action potential as a third phase, because it is very important. Already I said it will prevent the backward potential, also it will maintain the accuracy of action potential towards the next one. Okay, so this is very important. So some scientists consider that hyperpolarization is also a part of action potential. But actually, action potential means the potential energy will be in action. Okay, so that truly action is basically depolarization and repolarization. Depolarization and repolarization. This is the truly action potential, because this potential energy is in action. But here, below the resting potential, if you consider as a you are in a resting state, that means you are taking rest. But if you go below the rest, then you will go for the sleep. Okay, so when nerve will be in some extent it will inactive, that is hyperpolarized state. So it's a very that is for very few millisecond. That is the another issues, but hyperpolarization. So you can consider hyperpolarization is also a part of action potential. But truly, action potential means depolarization and repolarization. Okay, this is the action potential. Okay, so now I will come to the another point regarding action potential, that is refractory period. Okay, so through this figure I will explain, that will be very easier. So just I am erasing the another part, and I will explain the refractory period. So what does it mean, refractory period? That means you can think, suppose you order someone to do something, and he or she starts his work or her work, and at the very beginning you again order another work to do. So is it possible at a time to do things to be done by someone? It's quite impossible, almost. Okay, so your nerve cell just working like that. When it will be stimulated at the optimum level, that will reach the threshold level and it will start action potential, at that time if you just stimulate second time, nerve cell will never respond. If the stimulation is so excessive, so high, that does not matter, nerve will never respond. That is one refractory period. Refractory period, you can say nerve is in rest, nerve will not respond. Okay, so that period, very few milliseconds, that is not matter, but it is very important. So successively, repeatedly, if you just apply the stimulus to a particular nerve cell, then when it will reach the threshold, it will start its work and it will complete one action potential, then it will take another one to give its response. That means at a time the second stimulus will not work. So this is one point. So that is already I mentioned, that is the already mentioned in red mark, that is red color. Okay, that means from the threshold potential depolarization to the repolarization to the next resting potential, the true action potential state, depolarization and repolarization. In that stage, second stimulus will not work. Okay, so this period is very absolute, that is absolute refractory period. So refractory period means, already I said, in that period nerve will in generally will not give any response after taking stimulus. Okay, so absolutely it will not work. If you apply very strong stimulus, so no response. This is the absolute refractory period. This one, that means from threshold to peak and peak to resting. That is depolarization and repolarization. In that state, the nerve will never take any stimulus or any response in that period. So this is the refractory period. This is absolutely refractory period. And from the below the resting membrane, that is hyperpolarized state, and to the next, this part. Okay, that means in this region. When the potential energy will just go beyond the below the resting potential, that is hyperpolarized state, at that time if you apply the second stimulus, in generally it will not give any further response. That means from this part it will not create second action potential or second potential changes. That will not basically happen, in generally happen. But in that point, if you apply a very strong stimulus, then further action potential may create. Okay, so that means it will depend on the intensity of the stimulus. So here, in generally, response will not occur, but it will depends on the stimulus nature, that is strong or weak. So very weaker stimulus, it will not work. And even in such stimulus that will create action potential, if you just apply the same stimulus in that point, it will not reach to the threshold, because it will be needed, the stimulus is that will happen more than threshold potential, because already the potential state is below resting. Okay, so here the second stimulus, the response is usually not possible but not impossible. Okay, so this is the relative refractory period. Okay, so you can say from this part to that, this is the refractory period. Okay, that means you can say depolarization, repolarization and hyperpolarization, that combinedly in that state, basically nerve will not respond in the second stimulus. But if you just divided this to time frame, then one part is absolutely not, that is the absolutely refractory period from depolarization to repolarization, and during the hyperpolarization, that is another refractory period, that is relative refractory period. Okay, so this is the overall action potential related issues. Okay, so already I said membrane potential is different than action potential. That means your potential energy will be in action. So that means it will conduct, it will propagate from one region to another region. That is the conduction of action potential. The next discussion will be on conduction of action potential. Okay, so come to the point. So I'll just go on that. So what would be the conduction pathway of action potential? How it will conduct from one part to another? So if I just I want to discuss this, then you have to have a basic of a neuron structure, and already I said different types of ion channels. So in which part of the neuron, what type of ion channels are there, and how it will work, and how it will travel, that would be very helpful for you. Okay, so come to the next point or final one, that is conduction. So if I think in that way, if I just draw, then here this is like an ideal neuron. Okay, I'm not a good I have not a good hand in drawing, so like that. So this is your neuron or nerve cell. Okay, so this is the dendrite, and this is the soma, and this is the hillock, and this is your Schwann cell, and this one is node of Ranvier. Okay, and this is the nerve terminal. So this is the ideal neuron. Basically, the action potential will travel very quickly through this type of nerve cell, because just here I want to mention the nerve cell has basically two types. One is myelinated nerve. You already I said this is the Schwann cell, then this is the myelin sheath. Okay, this is the axon. This is axon. So this is the myelin sheath. Myelin sheath means this is made up of a lipid in nature, and some proteins are there, but basically lipid. So it will act as an insulator. Okay, when action potential will travel, it will not be influenced by any external effects. Okay, so how and where action potential will create? So here the dendrite. Through this dendrite, your nerve cell will receive the information, and within the soma has some nasal bodies and some other organelles, and that will process the information, and that will just push backward to the hillock, and then it will travel to the terminal. So which form the information will travel? That is, electrical flow, and another one, the nerve terminal had some synaptic vesicle, and it will carry a particular neurotransmitter. Okay, so neurotransmitter, the chemical messenger. So to just cause the exocytosis, to cause the release of neurotransmitter, action potential, that is electricity, is a must. So where electricity will create? So I will just remember, already I said sodium channel, the touch sensitive sodium channel, voltage gated sodium channel, leaky sodium potassium channel. Leaky potassium channel is very rich. Then I said sodium potassium ATPase or sodium potassium pump. I said voltage gated sodium, voltage gated potassium. I said ligand gated ion channels. Okay, so where they are? Basically, all the channels are there in dendrite and in soma, all types. But here, the voltage gated channels are very less amount. The soma and dendrite, voltage gated channels are there, but very less extent. Okay, so all types of channels are there, and over there dendrite and soma, ligand-gated ion channels are very prominent. Ligand-gated ion channels are very prominent and voltage-gated channels are very less. Where? The soma and dendrite. Okay? And hillock, this is the trigger point of the nerve cell. Very sensitive. And there, voltage-gated sodium channel is very prominent, and voltage-gated potassium also. So all other channels are there. The sodium potassium ATPase, that is the which is responsible for maintaining your membrane potential, is also present. Other ion channels are there, but over there in the hillock, the trigger zone of the nerve cell is highly prominent in sodium, voltage-gated sodium channel. Okay, and there the node of Ranvier, that means you can say the gap between the Schwann cell. Within the gap, very prominent in voltage-gated sodium and voltage-gated potassium. The leak channels and sodium potassium pump are also there, but ligand-gated ion channels are not prominent there. Okay, already I said ligand-gated ion channels are very prominent in dendrite and soma and also in the nerve terminal. Okay, but here the node of Ranvier are basically rich in voltage-gated sodium and also voltage-gated potassium, and some pump are there, but sodium voltage-gated is very prominent. Okay, so what will happen if you just touch the nerve, or it will just give the stimulus of the nerve? It can at a time you can stimulate the nerve cell in a higher extent that can create the threshold potential within the hillock, and it will create the action potential. Or you can touch here, and that will accumulate and it will reach the threshold. That means the accommodation, some summation of single or from that side, some extent of stimulus will aggregate and it will create the threshold level. That is possible. Or also you can like the punch in a strong way, and that will activate the nerve cell. Okay, so what will happen? So over there, action potential can be possible. In general, action potential is not generating there, because exactly the action potential initiate the hillock, because their voltage-gated sodium channel is very rich. But over there, in some extent that can happen, but usually not happen. So here, all the some sub threshold potential or graded potential will accumulate, and that will activate the hillock point. Then what will happen? Over the hillock point, the voltage-gated sodium channel will open. And then what will happen? Sodium will enter. In the resting membrane potential outside, sodium is very rich, sodium is very higher than inside, so sodium will enter. And then when sodium will enter, then what will happen? It will create depolarization. So already I discussed depolarization. So depolarization happen by the entry of sodium through the voltage-gated sodium channel, and instantly it will travel, because when sodium will enter, the intracellular sodium will travel through this hillock point to the next node of Ranvier. Sodium will just travel, and it will create the stimulus here, and that will trigger, or that will activate that node of Ranvier region, because in the node of Ranvier, already I said, voltage-gated sodium channel and voltage-gated potassium channel there. So what will happen? In which extent the potential change will happen through this inside, when depolarization happen in the hillock, the excessive amount of sodium will enter, and that will cause depolarization, and internal sodium that will travel to the next node of Ranvier, and it will activate or open the voltage-gated sodium channel. And here the node of Ranvier, the voltage gated through the voltage gated sodium, again sodium will enter. Okay, so what will happen? Again sodium will enter, then here again depolarization will create. That means exactly action potential will generate in the halo, and here action potential will regenerate. Okay, again here the internal or intracellular sodium will travel to the next node of Ranvier, and here the sodium will again enter. Okay, so here also again another potential changes, action potential. Here's another action potential. Okay, so action potential will travel very quickly as like jumping. Okay, because internal sodium will travel and some membrane potential will change, and that will combinedly will just activate the voltage gated sodium channel at the node of Ranvier, and that will create action potential, that is regeneration of action potential. So from this part to that part, again that part to that, it will travel towards the terminal. Okay, as like jumping. So this is called saltatory propagation of action potential, or you can say saltatory conduction. And this is happening very quickly because here no need to just maintain the resting potential. Just when sodium will just reach to the node of Ranvier, and that will open the voltage gated sodium, and sodium will rush into the nerve cell, and that will create the depolarization. And already I said where there depolarization will happen, instantly repolarization will happen. That means it will again try to reach to the resting membrane potential. This is the nature, okay, of the membrane, that will always maintain to resting membrane potential with the help of sodium potassium pump. Okay, that will happen, but actually through the depolarization, action potential will travel. Depolarization, because the highly activation part of the nerve cell, that is depolarization. If you want to activate the nerve, you must depolarize the nerve. Okay, so actually truly the depolarization is the main part for the activation of the nerve, that is action potential. Okay, so when the depolarization happen, instantly repolarization happen. Okay, so where depolarization already I mentioned several times, depolarization and depolarization, but after depolarization over there repolarization also happen. And how repolarization happen, already I said repolarization will happen by the exit of potassium, that is the repolarization. Okay, and it will maintain the resting membrane potential again through the sodium potassium pump and leak channel also. Okay, so basically there when it will travel, no hyperpolarization in a high extent, very little extent hyperpolarization happen. Very quickly depolarization will travel towards the terminal, and repolarization happen after that. Okay, so through this way, the nerve, through this axon, through the nerve fiber, very quickly electricity will travel towards the terminal. And then what will happen when the nerve terminal, the action potential will reach to the nerve terminal. The nerve terminal will contain large number of sodium channel, voltage gated calcium channel, like that. Okay, so when the electricity will reach to the terminal, it will, here is the calcium channel, and calcium will enter through the calcium channel, and it will bind the synaptic vesicular surface calcium binding protein, and it will cause exocytosis, and it will release the neurotransmitter. Okay, that is the chemical messenger, and that neurotransmitter will just bind to the next, that is postsynaptic terminal of the nerve or effector organ, as you know. So my part is action potential, so I am just coming back to the action potential. So this type of action potential is very common because That type of neurotransmitter, that type of neuron is very common because this is the myelinated nerve. Okay, but in our body, some unmyelinated nerve also. That means no myelin sheath. You can say this myelinated nerve as like electric wire. Within the wire, the copper wire there, and cover is like plastic cover. Okay, so some nerves are unmyelinated. There is no plastic cover, only copper wire. Okay, such type of neuron, you can say some pain conduction, pain sensitive nerve. You can say thermal sensitive nerve. You can say some sensory nerve, which are basically uh, unmyelinated. And that unmyelinated nerve, if you think like that, no myelin sheath, no myelin sheath. Okay, just I'm drawing here. So this is the halo, and this is like that. Okay, and throughout the nerve, the sodium channel, voltage gated sodium channel, potassium channel, sodium channel, potassium channel. Is there any gap? No, very continuously the channels are there. Okay, so what will happen when the halo will reach the threshold potential to generate the action potential? It will travel, and here action potential will create, here action potential will create, each and every channel will create action potential. And to maintain the resting potential, some energy will be required. Okay, so to restore the action membrane potential as well as to travel from hillock to the terminal, it will take more time. Okay, so this conduction is very slower, that is continuous conduction. This is very slow and it will take energy, and the saltatory conduction is very quick, and the continuous conduction. Okay, this is very slow, and saltatory conduction, you can say this is jumping conduction, very quick conduction, or propagation of action potential is very quick. And unmyelinated nerve, the action potential is very slow, and it will require energy, that will consume energy to restore the membrane potential for each and every part. Okay, so already I discussed the action potential generation, that is depolarization and repolarization. So just if you can think as like as saltatory conduction, that is conduction of the nerve. The conduction means, I said, generation, that is depolarization, that means it will create by the entry of sodium. Repolarization means it will create by the exit of potassium. So this is the same way, but the as here no myelin sheath, no node of Ranvier, so it will continuously happening, and it will create action potential that is very slow, and it will require energy. So in saltatory reaction, no energy required, and it will happen very quickly. So this is the action potential. So I'll just sum up the action potential. The membrane potential is always polarized in state in each and every cell, but when it will be functioning, it will just functioning means membrane potential is, also has some function to activate the intracellular signaling pathway like that. But when it will just travel in some particular muscles like skeletal muscle, smooth muscle, your cardiac muscle, and nerve cell, that will travel, and that will basically, the membrane resting membrane potential, threshold potential, peak level, that is equivalent sodium level, equivalent potassium level, hyperpolarized state, like that. So overall, after stimulus, the optimum stimulus, the if the potential changes will reach to the threshold level, only in that case action potential will generate, and before that if you just touch the nerve and it will just slightly open the touch sensitive sodium channel, it will create some potential changes, but it is meaningless because it will be under the threshold level. So it is called graded potential or sub-threshold potential. When it will activate the nerve, you will activate the nerve in a certain extent that will reach to the threshold level, then real action potential will create. That in that point, voltage gated sodium channel will open and sodium will enter. And through the sodium entry, the current will just go towards the positivity. Okay, so that will reach the plus 30 or 35, in maximum extent 55 in some highly stimulus state. And then at that point, peak point, sodium channel will close and potassium, voltage gated potassium channel will open. And through this potassium will exit and it will create a repolarization. And that repolarization will reach to the resting membrane potential. Again, this is called repolarization. Okay. After that, through the leaked potassium channel, some potassium will continuously exit and it will create the potential changes below the resting membrane potential, and that is called hyperpolarization. Okay, and after hyperpolarization, the nerve again will reach to the resting membrane potential, and that is done by the sodium potassium 80 phase pump. Okay, in that way, the whole action potential will generate. So truly action potential means depolarization and repolarization, and you can consider as third phase of action potential, that is hyperpolarization. And already I mentioned the importance of it. Okay, and in that way, depolarization and repolarization, the nerve through the nerve cell action potential will travel in myelinated nerve. It will travel very quickly as a jumping way, and unmyelinated nerve, it will travel slowly because frequently action potential will generate and it will travel very slowly and it will require energy. Okay, so action potential is very important. If you consider, already I discussed regarding the nerve cell, so neurotransmitter release action potential is important. Without action potential, nerve cell terminal will never release neurotransmitter. That is electrochemical signal. Okay, so electrical and chemical. But if you think about cardiac muscle, no action potential, no heart muscle contraction. If you think about skeletal muscle, not like that. If you think about the blood vessel, contraction and relaxation is needed. So such type of function, if you want to get, action potential is important. So some extent in nerve cell and cardiac muscle, action potential mechanism is somewhat different, but the basic thing is same. Okay, so this is the action potential, whole action potential in a row. So thank you very much.